Okay, all right. So, a very good morning to one and all. It gives me immense pleasure to welcome you all to this webinar organized by Department of Zoology of Dombili Shikshan Prasarak Mandal's KV Pendarkar College Autonomous on breast cancer on a chip, a paradigm for studying breast cancer physiology for therapeutics by our well-known resource person, Professor Goldie Oza, Professor in Laboratory of Micro and Nanofluidics, Mexico. Now I invite Dr. Mahajan, Head of Zoology Department at KV Pendarkar College, Dombivli, to give the welcome address.
Thank you, ma'am.
Mahajan, sir. Yeah.
Good morning to one and all. On behalf of Dombili Shikshan Prasarak Mandal's KV Pendarkar College of Arts, Science and Commerce Autonomous, I, Dr. Mahajan, extend cheerful welcome to our today's speaker, Professor Goldie Oza sir from Mexico. I am glad to welcome our principal, Dr. Suryakant Lasuneji. I also welcome Senior Vice Principal Sri Kishore Palak sir, Vice Principal and IPSC Coordinator Sri BT Sisat sir, Vice Principal Ms. Ashwini Bakkar madam, other staff members, my colleagues and student friends. I am happy to share that Professor Goldie has given consent to be a member of TYBSc Zoology Syllabus Framing Committee of our Autonomous College. This association with Dr. Goldie sir is going to benefit students at large. It gives me an immense pleasure to arrange this innovative talk on new trends in cancer therapy. Cancer in later stage called metastasis, that is spread of oncogenic cells to the distant organ, is very challenging to control it. It is necessary to spread awareness about the preventive strategies of this dreadful disease. Many people really don't know that malformed tooth can even cause cancerous growth due to the continuous physical irritation of tongue. Eating more vegetables only could also cause injuries to the intestinal mucosa as cellulose remains undigested. It may also cause intestinal cancer. I hope that such virtual platforms satisfies the objectives of cancer awareness among the society. I once again welcome all and thank you all for joining this Zoom platform to make this event fruitful. Over to you, Dr. Nisha ma'am.
मॅम अनम्यूट करा तुमचा आवाज येत नाही.
Yeah, thank you, sir. That was wonderful. Now I invite our respected principal, Dr. Suryakant Lasune, sir, to share a few words on this occasion.
Am I audible?
Yes, sir.
Yes, sir.
Good morning, all of you. Respected today's speaker, Goldie Oza, organizer of today's webinar, Professor Mahajan sir, Vice Principal, and also all my colleagues and students. I am happy to be a part of this event today, where in a chronic disease like breast cancer is being discussed in today's webinar. The world is going to change rapidly due to advancement of sophisticated technologies. Medical field is not exceptional. Scientists are working day and night. We now in the pandemic. The cancer is a burning topic to discuss in today's world. We know the some of the figures in 2018, 9.5 million people died due to the cancer. In 2020, around 10 million people died due to the cancer. More than 25 to 35% the people, women's dead due to the breast cancer. There are so many people, more than 25% deaths we can find due to the cancer. What is the cancer? Cancer is the unregulated cell growth. Our Goldie's Oza sir will give the detail about it. There are different type of the cancer which affect in the part of human bodies, such as the skin, lung, blood cancer, breast cancer, so on. There are so many causes are there, but we can say tobacco is one of the cause. But utilization of the tobacco or not, we can find the cancer. So many reasons are there for the cancer. 11 lakhs cancer patients registered every year in India. The total death of due to the cancer is more than 7.84 lakhs, including male 4.13 and also the women 3.17 lakhs death we can find in India. India is the second biggest country killed due to the cancer is the killer. This is one of the diseases. In 2020, 17 lakhs new cases has registered and 8.8 lakhs death due to the cancer. Sir, we'll focus on the advanced technology, advanced medicines, and how the WHO work on the last pandemic and also the given guidelines about the cancer and also the pandemic we know everyone. Here I don't want to take more time. I would like to conclude here my 2 words. I hope that the day prove fruitful to you all. Thank you. Thanks very much.
Thank you very much, sir. That was indeed informative and motivational. The current lifestyle with extensive stress and poor food habits is a serious threat to health. The consequential events followed due to the poor lifestyle is one of the major causes of cancer today. The occurrence of cancerous conditions is very common in all age groups today. In this situation, a prompt detection of cancer at an early stage can be of immense importance. Also, a quick and effective treatment technique is also the need of the time. It gives me great pleasure to introduce our speaker for today, who has immense experience in this field. I now request my senior colleague, Lieutenant Dr. Varsha Naravde, to introduce our speaker for today. Varsha, ma'am.
Hello, am I audible?
Yes, ma'am.
Yeah,
Yes.
okay. Thank you, ma'am. It is my proud privilege to introduce today's eminent speaker of webinar, Professor Goldie Oza. He is currently working as a postdoctoral fellow at Mexico on a project entitled Synthesis and Characterization of Gold Ferrous Oxide core shell nanoparticles and their applications in biosensor and drug delivery vehicle development, funded by Conacite, Mexico. This Jay has done his graduation and post-graduation from Mumbai University, which is a proud moment for us. He did his PhD from N. Shankaran Nair Research Center for Nanotechnology and Bio-Nanotechnology in Botany, entitled Biosynthesis of Nanometal and or its Complex by Algae and Bacteria under the guidance of Dr. Madhuri Sharon from Mumbai University. His experience in the field of nanobiotechnology, medical nanotechnology, and environmental biotechnology is remarkable. He has an impressive citation indices, which is representation of his hard work in the research field. He has a book published under his wing and 4 chapters in his credits. He has 39 journal publications and 26 research papers presented in the national and international conferences. He was awarded by ICMR Junior Research Fellow in 2004 and recently awarded as Investigator Level 1 as per National System of Researchers, Konakai, Mexico. Sir was also involved in community extension as a Director of International Services for Rotaract Club for the year 2000 and 2001. He has received many medals in elocution and essay competition and was active participant in sports. Now, I request our guest speaker for today's webinar, Professor Goldie Oza, to begin the talk.
Thank you, Dr. Varsha Narwade, for this beautiful and great introduction of mine. And now presently, I think I'm really very sorry that I could not send you my introduction maybe. And now presently I'm associate professor in laboratory. It's like actually known as Laboratorio Nacional de Micro y Nanofluidica. Actually, it's a national laboratory for micro and nanofluidics. So, and very much thankful to Dr. Surikant Lasune, who has explained basically about cancer and given a beautiful introduction. He being the principal and having so many responsibilities, he took really great time to not only be here, but to devote to explain about cancer also. And thanks a lot to Dr. Sharad Mahajan. He has actually, it's because of him that I am here presenting in front of you all. And thanks a lot, Dr. Nisha, for inviting us all and giving a great speech. And I would like to also thank Dr. Jagdev, Kailash Jagdev, who is also part of this. So I am really very thankful to all of you and all the august audience who is actually sitting here listening to the meager speech of or the session of mine. I am just a drop in the ocean. So it's just a micro drop, nano drop, basically, micro and nano drop. So we are just dealing with this micro and nano drop of the ocean, which is known as cancer. And so with this, we can start. I hope everybody is aware of, and as per the discussions and as per the introductions given by all of you, all big connoisseurs of the field here, I think breast cancer on a chip, which is really a paradigm for studying breast cancer physiology for cancer therapeutics, and we are dealing with in this particular institute, we are basically dealing with development of chips. Now, what is a chip? What is a cancer on a chip? Basically, we are mimicking cancer from the patients in the models in the cancer chips, so that we can grow the cancer cells derived from the patients, and we can grow it outside the body, and then we can study different kinds of physiology within the body, within the chip, as well as we also study the relationship between the immune system and the breast cancer cells. So, and then further, we also study what kind of therapeutics can be given, what kind of diagnosis can be done, so that we can identify what is the problem going on, what kind of mutations happen in the breast cancers, because every patient is different. Even though there are two persons suffering from breast cancer, but they are totally different in terms of. Hello. Am I audible? I'm sorry. Am I audible?
Yeah, you're clearly audible, sir. You're audible as well as visible.
Thank you, ma'am. So this is in this particular session, we are basically dealing with how breast cancer develops, as well as what kind of therapeutics are playing an important role in terms of nanotechnology, as well as different kinds of drugs which are being given, because we are basically using chips for simulating the human body, and then uh, like delivering drugs into the chip first, and then informing the oncologist that what kind of drugs can be given to the patient. So as I mentioned, that it's dealing with precision medicine. So we are dealing with individual patients, and individual patient is totally different. And that's the reason with this aspect, we would like to work on the therapeutic side. With this, as Dr. Suryakant has already mentioned, and it was a great thing that we have like a great amount of cancer mortality, both in men as well as in women. That is like till now approximately 10.13 million people are suffering. So this much of mortality and morbidity is occurring all over the world. It's like in the year 2020, that is the number which is being existing all over the world till now. So and the number of and in men, the highest number of cases which is existing is in prostate cancer. But the prostate cancer is like it's existing for sure, but the development or the growth of the prostate cancer is comparatively slower as compared to lung cancer. So the mortality in lung cancer is very high, and that's the reason in men lung cancer leads to high death rate, but the occurrence of prostate cancer is higher in men. And parallelly, when we talk about women, then the breast cancer is something which is creating havoc, because the number of cases, the occurrences, as well as the mortality is really very high. Mostly it's also due to the hormonal imbalances. So that's causing a great amount of disease. So we are basically working on breast cancer. Then even we have started to work on uterine cancer on a chip. And these are some of the very important cancers, and the number of cases, especially in US, Canada, Mexico, the number of cases are very high. Breast cancer is like it's a huge number, and uterine cancer is also moving towards it. So that's the idea. We are basically working on that aspect. So I think 1 minute. Okay. Yeah. So then we are talking about 1 second. Yeah, it's here. War on breast cancer. So breast cancer is the second leading cause of death. So it's you can imagine here that we are dealing with something which is there is a transformation from a non-metastatic tumor to metastatic ones, and there are different kinds of cells which are existing. One is the fibroblast cells, differentiated cancer cells, mesenchymal cells, immune cells. So all macrophages, adipocytes, cytokines, all of them, they're playing an important role to be existing in the breast cancer. And the estimated occurrences of breast cancer is like about 1.7 million cases, but that was in 2012. But in 2020, it's approximately 2.5 to 2.7 million cases. That is the range approximately of the cases, as well as the deaths are approximately 700,000 deaths in 2020. So that the numbers are really increasing. But of course, the death rate is decreasing because of the level of immune therapeutics and the drugs which are being given. So because of that, the death rate of breast cancer is decreasing a bit, but the occurrence is very high. Then we were talking about the different kinds of types and subtypes of breast cancer. So we have luminal A, luminal B, M2, normal like, basal like, triple negative breast cancers. So these are different types of breast cancers existing. And basically, the most dreadful amongst all of them is triple negative breast cancer. Because this kind of cancer is even we are targeting to this towards studying the physiology of this kind of triple negative breast cancer. That is number one. And number two, what we are trying to do is we are trying to identify different kinds of therapeutics for this kind of drug, for this kind of cancer, because triple negative means there is no estrogen receptors, there is no progesterone receptors, there is no epidermal receptors. So none of them exist in triple negative breast cancer. And that's something which is dangerous because we have the drugs again targeted against estrogen or progesterone receptors or HER2, but we don't have anything which is like for triple negative, wherein there is none of these receptors are expressed. So then what we could find now is recently we could find is since these are estrogen, progesterone, and human epidermal receptors, these are the ones which are more female receptors. And if the triple negative breast cancers, they are not expressing progesterone, that means they must be expressing testosterone in large amounts. Those females must be expressing testosterone. And then there was a great discovery here, wherein we could find that the treatment which was given against prostate cancer for men, the same kind of treatment can be given to triple negative breast cancer treatment, like patients, because they overexpress testosterone receptors. And that's the reason we could relate the prostate cancer and triple negative similar in terms of receptors. And then we could have the similar kind of drugs, and the treatments are going very, very in a very huge manner. There is mortality in the cells. So this is what we are working on triple negative breast cancer. But of course, that's something like the death in such patients is approximately within 3 months or 4 months. It's very rapid. It's very metastatic, terribly metastatic. So to treat this kind of disease is a very big challenge in front of all of us. And that's what we are planning to work only on triple negative breast cancers and developing breast cancer on a chip at this period. And yes, there are different kinds of cell cycling. I mean to say there are different kinds of inhibitors, EGFR inhibitors, and glycolysis inhibitors and PARP inhibitors. So these are okay. What are these? First of all, these are the molecules which are responsible for replication of the cancer cells. And now when the replication happens, and that can happen when the replication is happening, the glycolysis is increased in a large amount, like the glucose requirement increases, then glycolysis increases, the acidity in the environment increases, inflammation increases. So all these processes are overactive. And that's for that purpose, we need to have multi-targeted drugs, like multiple inhibitors, like selective anti-inflammatory drugs and aerobic glycolysis inhibitors. And so these are the multi-pronged actions against the cancer cells, so that they are not only metabolically, like they are cut in terms of their growth, but they are also being attacked by anti-inflammatory drugs and different kinds of telomerase inhibitors also. So these are some of the actions of the drugs which are existing to nowadays in the market. So yes, so we are basically having Vivacisumab. Okay, what is Vivacisumab? Now, everybody must be knowing about angiogenesis, or even if you are not aware of, but angiogenesis is something wherein the blood vessels are developed and they will be multiplied. So the cancer tissue has got the capacity to have more and more angiogenesis. I mean to say blood vessels developed, because what happens if the number of cells are increasing, then basically what in the body, in the normal person, what we have is, it's basically one capillary corresponds to one cell. So one capillary corresponds to one cell. That's a basic fundamental. Like we have 3 million cells, and then, like, I mean to say, millions of cells and millions of capillaries will exist. So this Vivacisumab is, what is it doing is, it will be attacking the angiogenesis process in the cancerous tissue, and then it will stop the development of blood vessels. And if the blood capillaries are not growing, are not made, then the killing will be, like we say, that then the cells will be dying out of starvation. The cancer cells will die out of starvation. For that purpose, it was really very difficult. It becomes very difficult for them to grow. And there are many other kinds of drugs like Siltuximab and Ruxolitinib, and then CD40 and then of course, now these are the drugs which are they are anti-IL-6. Now IL-6 is an inflammatory molecule. So when there is an inflammation in the body, then the cancer tissues, they survive because of inflammation. Most of the time, people say that, I mean to say, the scientific community, they say that just taking an aspirin will help treatment of cancer. So that's, of course, that's a very, very fundamental and the basic idea. But then they need to be given certain drugs and anti-immune system drugs. Anti-immune system in the sense that they should, like, there are certain macrophages which act. There are two kinds of macrophages basically. One kind of macrophage is helping the cancer cell, and the other kind of macrophage is known as positive macrophage, which will help our own immune system. And the negative macrophages, they will help the cancer cells. So it is all the game or the plan of macrophages basically. If the positive macrophages, that means macrophages which help the immune system, if they are dominant, then the negative macrophages will go down, and the positive macrophages will be leading to the death of the cancer cells, it will help in the killing of the cancer cells. But if the negative macrophages move up, and then they are more dominant, then the positive macrophages go down, and then negative macrophages will help in evading basically the immune system. They will just tell the immune system that, come on, my friends, go back. This area is completely safe. It's a safe zone. So this is the message they give to the immune system, and that's the biggest problem in the body. So of course there are different kinds of drugs like nivolumab and ipilimumab and cisplatin, and all these drugs are responsible for chemotherapy of the like for cancer cells. So we are we were dealing with the different kinds of like core shell nanoparticles, and here we are talking about core shell. Means the core will be made up of iron oxide and the shell is made up of Au. And nowadays we are working on different kinds of shells like copper and then gold and then even platinum, palladium. So these are some of the molecules which we are using. But copper is the copper and gold. These are the ones which are very, very useful for the body. And that's the reason we are using gold or copper for covering the iron oxide or cobalt ferrite or zinc ferrite or nickel ferrite kind of structure. So we are also making different kinds of ferrites, and we are covering it with different kinds of metal nanoparticles, like I mean to say gold or copper nanoparticles. Yeah. And then this is the core shell. You can see here that we have this the core made up of iron oxide, as we have already seen, and then the shell is made up of gold. So this particular complex is really working very efficiently, and it gets inside the cancer cells. These are the cancer cells, and they are getting inside the cancer cells, and they are like I mean to say they are forming the endosomes, and then gets into the lysosomes, and they are just getting in and then slowly and steadily, because of the acidity, they start releasing the drug, which are basically on the surface of the nanoparticles. So this kind of work we are doing. Of course, this kind of work is it's not that great, of course, but then but we are getting some idea that how that these nanoparticles are really getting inside the cells, inside the cancer cells, and what is the mechanism of the nanoparticles getting inside the cancer cells, and how do they kill the cancer cells. So these mechanisms we are trying to understand in details. So and then on the right hand side you will find this is a squid, which is basically telling about, it's a magnetometer, which is telling about what kind of magnetization exists in this kind of complex system. So you can see here the magnetization is approximately 80.9 EMU per gram. So this kind of this 80.9 EMU per gram is something which is very very high for the killing of the cancer cells. Why? Because these so why do we use magnetic nanoparticles? First of all, the idea is when we are using magnetic nanoparticles, so magnetic nanoparticles are responsible for generating hypothermia in the body and in the cells, in the cancer cells as well. So they will be so what we have to do is the magnetic nanoparticles get into the body. They will be injected into the body, and then the whole body will get into magnetic resonance imaging system. And once they get inside the magnetic resonance imaging system, then there is a radio frequency which is focused in the region wherever there is a tumor. And then after that, there is these radio frequencies which will be responsible for the activation of the magnetic nanoparticles, and then these magnetic nanoparticles will be vibrating. And then due to the vibration, there is a heat generation, and that vibration is due to the Brownian fluctuation. Basically, I think I'm telling in very like I don't want to use any jargons here. So the right term is there is a vibration in the nanoparticles, and these vibrations lead to heat, and the temperature rises to 45 degrees Celsius, which is very high. Localized heating happens in the tumor. So that localized heating is approximately 45 degrees Celsius in very, very local region. And then that leads to killing of the cancer cells. And then what is the reason why we are using this gold? The reason we are using this gold is gold nanoparticles are easily trackable, number one. And the second point is they can be attaching drugs onto the surface. So drugs which are attached onto the surface of this core shell, they will be like it will be easier for the drugs to attach onto the gold nanoparticles, because these drugs will be like having nitrogen and sulfur on the surface, And then gold is having very high affinity towards nitrogen and sulfur surfaces, and that's one of the reasons why that we have used gold onto the gold and iron oxide. So gold is basically useful for attaching with the drugs, and core and the iron oxide is basically useful for inducing hypothermia. So that's the main motto behind this kind of core shell nanoparticles. So then you can see here, we have also used cobalt ferrite, and this is like a color, I mean to say, this is a flower-shaped structure. So the core is made up of cobalt ferrite, and they are all covered like in a form of a flower by gold. And this is a structure which is generating more heat. Cobalt ferrite is generating more heat as compared to iron oxide. So this is a basic idea why we are interested in using this. And then manganese ferrite, these are also responsible for the formation of this kind of flowered structures. So, of course, this complex nanoparticles are useful for basically treatment of cancer, and that too in small amounts. We don't need a high amount as compared to iron oxide, wherein we need little bit, the concentrations are a little higher. But in this, since the magnetization is very high, these particles can be useful for treatment of cancer in a very, very small amount of the nanoparticles in the body. And then we have this MRI and microwave-based hypothermia. So I mean to say, there is like magnetic resonance imaging. We can identify where the nanoparticles are going because of the iron oxide. You can see here, these are those dark places. So these are all images wherein I can easily identify where are my nanoparticles present. So this is basically seen in magnetic resonance imaging. And you can see here that 45 degrees Celsius temperature rise. So this happens in the hypothermia condition wherein the temperature rises very soon. So what we are interested in doing is how to, for example, we are sending the drugs, we are sending the nanoparticles in the body. So once we're sending the nanoparticles in the body, then how the body will recognize that, come on, these nanoparticles are supposed to go to cancer. They should go towards the cancerous tissue. So the main idea is we need to have a targeted version. So either we can have folic acid on the surface because folate receptors are overexpressed on the cancer cells. And that's the reason we have this nanoparticles having folic acid onto the surface along with the doxorubicin, drugs like anti-cancer drugs, and then we can just inject into the body so that folic acid will track the folate receptors and they will bind it. So and these folate receptors will be overexpressed onto the surface of cancer cells, and then it's easy for us to to have a targeted drug delivery. That's the motto behind using folic acid. It should not go anywhere else in the body wherein there is no folate receptors, because yes, folate receptors are also expressed by normal cells, but the normal cells will always have folate receptors which are always covered. They will not they will not express folate receptors all the time. They will be always covered. But in the case of cancer cells, since they are in need of all kinds of glucose, vitamins, everything, whatever cancers, the cancer cells, whatever you eat, they will eat the first. And then whatever is remaining, the body gets it. And that's the reason, the body survives. But it is like half dead. So there is muscle wasting in the body. Like all the muscles, they get degraded because they don't get energy. They don't get food. They don't get energy at all. And that's the reason there is no energy at all. So basically, this is the problem. All the cancer cells will take away all the nutrients along with the glucose and everything. And I think like cancerous tissue, they have a pH around cancerous tissue, the pH is approximately 6.7. It is little acidic. It's not neutral. In the normal tissue, the pH is approximately 7. But in the case of, I think, everybody can see my presentation, whole presentation?
Yes, sir. It is very clear. Yes, sir. It is
Okay.
pretty clear, and you are also very much visible. Sound is also very clear.
Okay, yeah, okay. Yes, thank you. So then this is like doxorubicin and iron oxide, what happen, and with folic acid, this whole complex, they will be released in large amounts. You know, first of all, they will be released in pH 6.7. They will not be released in 7.2. Or so normally the body has got 7.2 pH, but in the cancerous tissue the pH is approximately 6.7 because glucose is taken up by the cancer cells in large amounts, and they do anaerobic glycolysis. They don't do aerobic glycolysis in large amounts. They do mostly anaerobic glycolysis because they want to use glucose as much as they can. So there is a kind of anaerobiosis in the system. And that's the reason what happens is this, due to the anaerobic glycolysis, there is a large amount of lactic acid and of course, pyruvic acid first and then lactic acid formation happens. And due to that kind of acidity generated, there is good amount of, the pH goes down to 6.7. And in that particular pH, these nanoparticles, they have some pH sensors onto the surface. They will now get attracted towards those regions where the pH is less, and then they will be taken up by those cancerous cells. And further, when the cancerous cell nanoparticles with the doxorubicin, with folic acid, of course, everything gets inside the cancer cells, they get into the endosomes. And in the endosomes, the pH is approximately 5.3. And at that particular pH, there is like the complete release of the drug and the killing happens. So that's the basic idea of this kind of treatment. So you can see here 7.4 is approximately, 7.4. Basically, it is 7.34 to 7.42. This can be the range of a normal pH. And then, 6.8 is in the tumor tissues and 5.4 in the endosomes inside the cancerous cells. So this is the basic idea of how the treatment happens. Like the cells come outside they are just outside the cancerous cells, they're endocytosed, get into the endosome, and then they happen to be killed. And we were studying with different kinds of cells. One is the L6, like oral cells. It's a normal cell, and Hep2 is a carcinoma cells. So Hep2 is basically carcinoma, larynx carcinoma cells at this stage. So we just wanted to check what kind of how cancer cells are being treated using these nanoparticles. And we could find, yes, that the cancer cells are being killed in a large amounts as compared to the normal cells. So then we did a kind of confocal microscopic studies, and we could find. Then we have also done this kind of studies in triple negative as well. So triple negative breast cancer cells were showing a great amount of killing in the the killing based on apoptosis. There was an induction of apoptosis due to the nanoparticles, the complexes which we have added into the triple negative breast cancer cells. This is triple negative MDA-MB231. It's a triple negative breast cancer cell, and we could find that yes, the cancer cells are getting killed because of this complex. Then after that, we wanted to study that, okay, what is happening in terms of hypothermia and chemohypothermia. That means, for example, we inject so the idea is, to tell you in short, like we inject the nanoparticles inside the body. And then when we inject the nanoparticles inside the body, first line of treatment is like hypothermia. We induce a hypothermia in the body. So in a particular localized region. Once hypothermia is induced in the tumorous region, then after that, the drugs will be the complexes will start releasing itself. They'll be attaching itself onto the folate receptors. And then further, they will get inside the cancerous cells. Then once they get inside the cancerous cells, then the cancerous cells will be, then after that, the cancerous cells will release the drugs because of this high pH. The pH is 5.3 in the endosomes. So at that particular pH, the cancerous, the nanoparticles will release the drug. Okay, so what we are doing basically is we are doing multi-pronged or two-pronged action. One action is like we are inducing heat. Then second point is we are killing the cancer cells by the anti-cancer drug. So there are two kinds of things going on parallelly. And in future, what we are planning to do is, what we are intending to do is, we should have an immunotherapeutic drug also attached to it. And the fourth thing, what we are also attaching now is microRNA. So there are microRNA, then the anti-cancer drug, then the folic acid receptors, folic acids, which is targeting towards the cancer cell, and hypothermia. So all of them, are like it's a multi-pronged action wherein the killing, like if one doesn't work, the second works. If second doesn't work, the third works. If the third doesn't work, at least the fourth works. So if there is a multi-pronged action, then and then only the killing can happen, and that too, the concentrations at which it needs to kill the cancer cells is also like all the cancer cells must be killed. To that extent, we have to work on these nanoparticles as well as the microRNA or the drugs. So that till that extent, we would like to work. Then, of course, we were doing this flow cytometry, and through the flow cytometry, we could find that there is apoptosis induced. So this confirms that there is apoptosis induction in the cancerous cells. And then we were also studying the mice. So mice, in the normal mice, we injected these nanoparticles, everything, and we could find that, yes, there is like after 24 hours, almost after 24 hours, all the nanoparticles are being excreted out. Nothing happens to the body, and it's not accumulated in large amounts. But in the case of tumor-induced mice, wherein we had induced tumor, we could find that when we injected the nanoparticles, it stayed there for like till 24 hours, and it stayed there till 48 hours as well. So in this case, we could find that till 48 hours, these nanoparticles were still there inside the body in the tumor-induced mice. But in the normal mice, you will not find this. Basically, everything goes out in 24 hours. So this is the basic difference we could find when the nanoparticles are present, because basically cancer cells have got great affinity towards nanoparticles, and that's the reason. One of them, like all of them, they stay there. Yeah. And then we also induced hypothermia, and in the mice we could do. This is the probe. This is the probe which was inserted, and that probe will be responsible for radio frequencies. And those radio frequencies, when like they will be moving like a wave, and then since the nanoparticles are already there in that tumor region, due to the waves, these nanoparticles they get decreased. I mean to say, sorry, the tumor size decreases, and as the tumor size decreases, the survival rate of this mice increases. So this was a very small experiment which we performed on mice too. But then we also wanted to perform that. Okay, what happens to the immune system? We wanted to study on mice, but we wanted to study before mice. We wanted to study on transwell, 2D transwell systems. So this is a 2D transwell system. So wherein we have this upper compartment wherein there is 1 second, there is some issue in this. Okay. All right. So on the upper compartment, there are macrophages. And as I had mentioned, there are macrophages on the upper, there are two kinds of macrophages. One is positive and the other is negative. So we wanted to study, okay, come on, what happens? What happens when the nanoparticles are added, like in the lower compartment, we added the cancer cells, and on the upper compartment, we added the macrophages. And we wanted to check, and then we also added the nanoparticles in this region, in the region wherein there are cancer cells growing at the bottom. So we could find that all these macrophages, they travel through this porous membrane. They are traveling through this porous membrane and they get out of it. So once they are getting out of it, then what happens is, I think, I want to use, can I use a mouse here? Because I think I don't have permissions. Okay, wait. All right, not a problem. So here we have, on the upper compartment, we have the macrophages, and the lower compartment, we have the cancer cells. And as these nanoparticles are grown in, I mean to say, they are interacting with the cancer cells, they will attract macrophages towards them. And these macrophages will traverse through this porous membrane, and then they will start multiplying in this region, and they will start killing the cancer cells. So this kind of study we wanted to perform using this 2D transfer system, and we have done this kind of study, and we could find that the macrophages, like the positive macrophages, they were effectively active, and the negative macrophages, they became inactive after a certain stage. So that's what we wanted to understand in this kind of work. Then we were interested in doing some personalized approach kind of work, that, okay, breast cancer, how it works, how many days it takes and to grow the breast cancer cells and all those things, and how much time it takes for the cancer cells derived from the patient biopsy, how much time it takes to grow the cells. And not only two-dimensional, we wanted to grow them in three-dimensional form. So then we started working on the three-dimensional system. Why? What is the reason? The reason was that most of the time, as you have seen this. This is a poor mice. I'm saying it as the poor mice, but I'm really feeling bad about it. That it's a nude mice, immunocompromised mice. Of course, after this experiment, we had to kill it. So now in this situation, you can imagine that we are becoming so much, and of course, you're all zoology students, most of them. So it becomes very common for the students of zoology and physiology, and it becomes very common, and even for pharmacologists, for any kind of person working on drugs and nanoparticles, and also different kinds of pharmacogenomics, everything, metabolomics. So people have to work on mice, and then after that, they have to kill them. So, and then they simply take out the organs, and they start doing the microtomies and do all kinds of studies with that. So we wanted to avoid that. And of course, the whole world wants to avoid that. They don't want to kill mice just for the sake of, for our survival, for our enjoyment, basically. That's happening. So, yeah, because for our survival, right, we are testing cancer drugs on them, and that's really not good on our part. So then what happened was we started working on three-dimensional system. Okay, what's the difference between mice and the human, human system? The basic difference, very basic difference is that the human system, the markers, the cell surface, and the physiology is a bit different as compared to mice. So what happens is whatever studies, whatever drug studies which we are doing on mice cannot be imitated, cannot be seen. We won't be getting same results in terms of in mice as in human beings after that. And that's the reason most of the drugs which are being developed, like the drug discovery happens, and like there are millions of drugs which passes through phase clinical trial 1, 2, 3, and 4. After that, after so many drugs, they are like being removed from the clinical trials, and at the end of the day, we are left with only 1 or 2 drugs, and that's something unfortunate for the scientific community. So that's the reason we have come up with this kind of three-dimensional system, wherein three-dimensional we are growing the cancer cells in three-dimensional form, not in two-dimensional form, because two-dimensional means the cells are confined in 2 dimensions, and they are not exactly mimicking, because the body wherein the cells are there, they are always growing in a 3-dimensional form. They're not growing in just the 2-dimensional form, and that's the reason this kind of 3-dimensional system is very useful for studying the physiology in a very accurate manner. So this was like from 2010 to 2017, people have developed this kind of systems like breast cancer on a chip. So then what we have done is we started working on one kind of system, and that's known as spheroids. Now, what is a spheroid? Spheroid is basically a 3-dimensional structure. This is in collaboration with Dr. Alejandro Centella, who is one of the medical doctor here. He's an oncologist, as well as he's a researcher in a medical institution here associated with British American Hospital. So what happened was this spheroid development is something wherein we were interested in growing the cells in a 3-dimensional form, and they are not regulated form, they are unregulated growth. Of course, as cancer is growing, here we are also growing the cancer cells in an unregulated fashion, and we get at the end of the day a clone or a tumor. Now this kind of tumor has got all the different kinds of cells. It has got as I mentioned, it has got a stem cell, a differentiated stem cell. It has got a immune cell, everything mixed in it. So this complex, this tumor, which is grown outside the body, not inside the body, they're grown outside the body in a 96-well plate. So what we did was, in collaboration with the medical doctor, we got the patient's samples. And then from the patient samples, like the breast cancer samples, we just grew the breast cancer cells in the form of a spheroid. So what is a spheroid? It's just one single cell or 2-3 cells. They will be added into the medium, DMEM medium, and then they will just then we'll be putting some growth factors into it, and once the growth factors are added in a specific way, then these cells, they will start multiplying, and then they form a very small tumor. So in one single well of that 96-well plate, one single well, we will have a good amount of triple-negative breast cancer or breast cancer cells in the form of a tissue. Now, in this tissue, we are just injecting the drugs, and we'll test which drug is more effective. And once we find which drug is more effective, then we can tell to the doctor, come on, these are the drugs, doctor, this is the concentration, and this concentration is effective for this kind of cancer. And for this kind of cancer, okay, there is breast cancer luminal A type. This is breast cancer luminal B type. So luminal A type, you can give doxorubicin. Luminal B type, this I think etoposide is working fine. But if there is both luminal A type, both the there are two patients, both of luminal A type, then what happens is most of the time, even if we give doxorubicin to both of them, none of them works. Then we have to test, okay, for this kind of luminal A type, this cisplatin works well. But for this kind of luminal A type, paclitaxel works well. And then what is the concentration? We have to give that concentration and then check. And then easily we will find out what is the minimum concentration of the drug which can be given to the patient so that the cancer cells are being killed. But at this moment, a kind of randomly doctors are doing the job, wherein randomly a patient comes, okay, luminal A type, all of them, let's give doxorubicin or let's give carboplatin. That's it. So that's the thing going on presently. So, but with this breast cancer on a chip model, we will be able to find out which drug can be given and what should be the concentration of the drug. That's the basic motto. So what we could find is this is a spheroid, and we added trypan blue. So when the trypan blue is added, you can see here without any treatment, and this is a negative, and this is a treated sodium azide is added into it, and this is control positive. So this confirms that there is killing of the cancer cells. There is apoptosis developed. So I mean to say, so these are the two drugs which are effective against this kind of cancer. One is carboplatin and doxorubicin. So this confirms that these two drugs are very effective in the killing of the cancer cells. So we can also do a comparison that, okay, in 2D, which drug is working fine, in 3D, which drug is working fine. And based on that, we could find, okay, in 2D, some other drug is working fine, but in 3D, some other drug works fine. So that means based on this comparison, we can easily identify that, oh, 2D doesn't work very effectively. Three-dimensional cells and systems are working very effectively in identifying which drugs can be given to the patient, because it's exactly mimicking the tissue. Then we have done then we could find this is a three-dimensional growth of cancer cells. So there is a so these spheroids. Of course, this is a spheroid means it's a complex culture of mixed culture cells of breast cancer and immune cells and everything. But basically, it is 95% what we are doing is we make a spheroid out of only cancer cells to have more control into the system. That's the idea. So we have this kind of spheroids grown on in the three-dimensional microfluidic system. It's a microfluidic system. And then then there is a stromal cell. Stromal cell if you see in the body, the stromal cells are giving support to the cancerous cells or to any kind of cells. Then these stromal cells are made up of fibroblast, and there is a lower channel. And these lower channel are basically, they are responsible for supplying some heat, some nutrients to the medium. To the nutrient medium is supplied to those microfluidic system. So this is the way you're growing the cells as well as the tissue. And I think, so when there is growth in the microfluidic system, then we do confocal microscopic studies, and then we can find out easily whether the spheroids are grown or not. That's the idea. So due to the color maps, we can easily identify whether the spheroids are really growing or not. So it's like day 1, day 2, and as the number of days increases, we can find the number of cells are growing effectively. And that's the main idea for this kind of work. Then we have to give some kind of treatment to this kind of tissue. So in day 1, there is some kind of treatment. Day 2, there will be little cytotoxicity. Day 3, there will be more cytotoxicity. Day 4, there will be very high cytotoxicity. So this way we can identify that, okay, this particular drug is really effective or not. So in 7 days period, we can find very high toxicity in the cells, and that confirms that, yes, this particular drug is very effective for the patient. And then we were also studying, okay, what kind of matrix, because in the body, there is an extracellular matrix like proteoglycans, collagen, based on which the cancer, on which the cells are growing. So similarly, we also need in the chip, we also need what kind of gel or extracellular matrix is required so that these cancer cells can grow very effectively in the 3D system. So we have this matrix gel. Matrix gel is basically a gel or hydrogel. It's just a gel. You can imagine like agarose gel, right? Everybody knows agarose gel. So it's basically a kind of agarose gel, but this agarose gel has got all the nutrients into it, all the kinds of growth factors, whatever you need. And then you add the cancer cells onto it, and then due to the growth factors, the cancer cells will start multiplying and multiplying. And in 7 days, you can easily find out that how much growth has happened in the cancerous tissue. So this is like the difference between this 2D and 3D. Like we have in 2D and 3D, we have added the drugs, and we have found which drug is more effective in 2D and which drug is more effective in 3D, and we could find that, and then we give to the patient and we confirm, hey, come on, this olaparib was really good for in 3D system. And then when we give to the patient, yes, there is a good amount of cytotoxicity in the cancer cells in the patients, and they are showing good results. So that's the motto behind doing this 3-dimensional system. So what we have done is now what we are doing in our lab is basically developing this kind of 3-dimensional structures, of course. But you can see this is one channel, this is second channel, and this is the third channel. So these 3 channels are basically responsible for like growth of like one of them will be responsible for the growth of cancerous cells. In this region, there will be cancerous cells. And then in the second region, there will be endothelial cells. Endothelial cells are mimicking the blood vessels. And the last one, this is the immune cell, this channel in which we are having the immune cells. All of them, they will be, you can see here, these are very, very small pores, very small pores. So that through these pores, only proteins or cytokines or interleukins can traverse. None of the cells can traverse through it. So what happens is, and what you can see this black one, this is basically the impedance spectroscopy. So this impedance spectroscopy, this is at the base. So at the base of the 3-dimensional microfluidic system, we have this impedance spectroscopic system, which will be responsible for detection of cancer cells. I mean to say cytotoxicity of the cancer cells. They will say the number, exact number. Hey, come on, there are initially the number of cells were 100. Now it is 50. Now it's going down to 20. Now it's going down to 10. So this impedance spectroscopy is giving us that numbers. And all the cells are, hello?
Yes, sir. Yes, sir. Yeah. Continue. It is clear.
Okay.
Yeah.
So, hello. I'm sorry. Could you want to say something? I'm sorry.
No, it is clear, and go ahead. No one asked anything.
Thank you.
Yeah.
I'm sorry. Yes. So this, of course, if there is any doubt, I can, of course, clear out whenever during the session of the doubt sessions. But this is a kind of prototype which we have developed for the development of the. So in this prototype, we are doing 2 jobs. One, sensing. In the medium, how much amount of glucose is added, what is the pH of the system, what is oxygen, how much amount of oxygen is present, and if the oxygen goes down, immediately the oxygen is made up into the system. So like that. So we are doing the sensing job, and we also measure the cytotoxicity in this. And second job is the growth of the cancer cells. That's the main motto of this. So I think this is, and of course the last part is we are also detecting, as I mentioned, we are also interested in detecting the cancer cells. So this is a nanovell cytometric system. This is in collaboration with the Massachusetts Institute of Technology, MIT, and Harvard Medical School. So my PhD student, he has done his 1-year work in MIT, and so due to this collaboration, we could have a good amount of work wherein this is a nanowell system wherein you can detect cancer cells in, so you can have one cancer cell or two cancer cells in one nanowell. So, and in each nanowell, there is a fluorescent biomarker which will interact with the cancer cell and then it will fluoresce. If there is no interaction, I mean to say, you can see here, there is no cancer cells, there is no fluorescence. And if there is a cancer cell, there is fluorescence. So this kind of system was developed by one of my student, Kalomari. So so you can see here, this is like a colorful image. It's a color graph wherein you can detect different markers on the cancer cells. For example, you want to detect overexpression of folic acid receptors or overexpression of nucleate or overexpression of HER2 receptors, overexpression of progesterone receptors or Herceptin receptors, any of these. So these will be individually detected, like each well will have one antibody specific against one receptor. So you add the cancer cell, and if that cancer cell is overexpressing for one particular marker, they will easily detect that. So that's the idea behind this kind of nano system. So I think with this, I would like to finish off my session. And here we have, I think I'm really very not on time. I think so. I've exceeded my limit, right?
No, sir, it's okay. We are fine with it. It is more important that we get more information like this. It's a golden opportunity for our students to explore new aspects. Thank you very much, sir. That was a very innovative and very brilliant talk. And I hope this talk motivates our students to explore and empower themselves to grow into research. It was really very thoughtful of you to share so much of information and knowledge with us.
Thank you. Thank you.
Yeah. Now the session is open for queries and discussion. So if anyone is interested, they can ask some queries or doubts in anything. Students or teachers, anyone who is on the participation list can go ahead. If they have any doubts or queries, you can ask. Sir, if you don't mind, can I ask one question?
Sure. Thank you.
Yeah.
Please.
Sir, sir, I was thinking that folic acid, which is used to as an identification for this as a marker. So is that coated on the nanoparticle or only the receptor will be coated on the nanoparticle? How is that?
Oh, no, only on the nanoparticles there will be acid, folic acid. Attached.
Okay.
Just the folic acid will be attached, like it's a vitamin, right? So folic acid will be attached onto the surface of the nanoparticle, so that this folic acid will interact with the folic acid receptor. So the cancer cells will overexpress receptors for folic acid.
Okay.
So this receptor is existing on all the normal cells as well. So they're all expressed on normal cells as well as on cancer cells. But generally on the normal cells, like for example, I've shown you that these are the receptors for folic acid receptors. But in the normal cell, it will be always like in the opposite side. They will not be opened. Whenever they need, they will be they have to open it like that for a fraction of a second. They will open itself. And then once they get the folic acid, then they will close it itself. So that's the mechanism of the transmembrane protein, the folate receptors. But in the case of cancer cells, generally this mechanism is completely ruptured, or I don't know what happens. There is a lot amount of studies which needs to be done on this aspect. But generally, the folic acid receptors will be overexpressed. Generally. The open receptors for folic acid on the normal cells will be approximately 10 raised to 1 or 10 raised to 2. I mean to say
Okay.
10 or 10 raised to 2 hundreds. That's it. But in terms of cancer cells, it is approximately 10 raised to 5 or 10 raised to 6. These many receptors will be expressed onto the surface of cancer cells. So you can imagine that how much amount of nutrients like folic acid they will be gaining. So
Yeah.
that becomes our interest of this is the way wherein we find opportunity in killing
Yeah.
the cancer cells.
That's a very interesting aspect over there, how
Yeah,
they
thank you.
identify.
Yeah, that's something really great. Yeah.
Yeah.
Can you
Okay.
please, I'm Mahajan. Can you please comment on the significance of mammography on this occasion?
This is actually the mammographic image is basically, okay, first of all, we would like to work on the mammographic images, of course, for sure. Because it's not my job basically, but it is the job of the oncologist, and they are basically doing the mammographic images. But my job, what we are doing on the mammographic patterns is we are applying artificial intelligence, like whenever we are doing a scan, whenever we get the mammographic scan patterns, then we are we are zooming it in and through the artificial intelligence, and like we can have 5Ds and 6D and 7, like 6 dimensions zooming in of the patterns, and we are trying to identify that whether there is any cells that, like there is any cancer cell existing, because most of the time in the mammographic images or the patterns, people are not finding the cancer cells, but using the AI, artificial intelligence, and the mapping procedures, which have been very enhanced nowadays. So now even once, like previously it was like the tumor size must be approximately millimeters, right? But nowadays they are in micrometers, or even in like even lesser than that. Then the number of cells are at least 50 or 25 to 50. Even that numbers can be detected nowadays. So that's something which is possible only through artificial intelligence, which is happening nowadays through for this mammogram.
Early stage treatment is possible because of mammography.
Yes, exactly. But the only problem is what we are like. Not only we, basically the patients. You know what happens is, unless or until the size increases to some millimeters, the patient doesn't understand. So, but nowadays, since the detection, I mean to say, detection happens, but there is every year there is screening of breast cancer screening going on every year. So based on that, people can easily get the idea that like even in small, very small amount of tumor cells exist and they can be detected. So using this AI and due to the breast cancer screening, this is happening very fast. Yeah. That's something which is very important.
Thank
for
you.
Sure. Thank you, sir. Thank you.
Is there any? Yes. Is there any cell size difference between malignant cell and non-malignant cell or normal cell?
The cell size.
Yeah.
Mhm.
Please.
The thing is, the cell size, yes, there is of course a difference, and moreover, the receptors are also a difference.
Yeah.
So the cancer cells, they are of course bigger in size, and moreover they are since they lose the attachment anchorage dependent mechanism, they lose that, so they are eligible to move. They move here and there, and they can easily come out of the blood vessels for sure. So that makes the cancer cells more, even they become more flexible. The flexibility increases, wherein the normal cells, they are not that flexible. but the cancer cells become more flexible. There are like the microtubulins and the tubulins, they are like completely, more flexible for them. And that makes the whole problem for us. And that leads to, because of this, metastasis is very easy for them. So these are some of the problems. Yes, due to the size and due to the flexibility, cancer cells are really creating problems and havoc in the system. Yeah.
Thank you, sir.
And moreover,
Dr. Mukherjee,
yes, sir.
I have actually 3 questions. Should I ask one by one or all 3 together?
As you wish, sir.
Okay. So one question is that you have said in between that cancer cells survive during inflammations and so on. Now, whenever there is an injury, the inflammation is basically caused by the release of the prostaglandins. So,
Yes.
can we modify and control the release of prostaglandin and thereby starve them of the growth? Number one. Number two is how much is the use of interferons in the research right now? And thirdly, we have one more technology, and that is introducing boron nanoparticles, which are absorbed preferentially by the cancer cells and then causing hyperthermia and killing them selectively. And to my understanding, at the moment, 3 such sessions with boron nanoparticle treatment is almost removing every cancer cells. So these 3 questions on you.
Sure. Thank you, sir. Thank you. Actually, number one, as you mentioned about the prostaglandins, and of course, not only prostaglandins, there are leukotrienes as well. So these are some of the these are the people which are responsible for inflammation. And I mean to say, these are the consequences of inflammation. And of course, whenever a person is taking any drug, anti-inflammatory drug, NSAIDs or AIDs, whatever. So these drugs are basically affecting them, the prostaglandins and leukotrienes. So whenever nowadays, like, because after the I mean to say in the year 1998 or 2000, I think so, from that particular years, people have started giving anti-inflammatory drug as well, apart from the anti-cancer drugs, chemotherapeutic drugs. They're also giving the anti-inflammatory drugs as well, because they, of course, because of and moreover, biopsies, they are also responsible for creating injuries, as you have mentioned it rightly, that as soon as the injury happens, then that leads to inflammation, and inflammation means it helps in cancer. It helps in the growth of the cancer cells for sure. And nowadays, what people are intending to do is they are doing liquid biopsies. They're not interested in doing the biopsy of the tumor. They don't want to even dare to touch the tumor, because people generally till even now, people are in undeveloped countries, or developing countries, I should say, sorry. So in the developing countries and in most of the countries, people are doing biopsies still. The solid tumor biopsies are happening, and then that creates the inflammation in the tumor region. And as soon as the inflammation happens, that benign tumor becomes malignant and metastatic. The whole process immediately starts. So yes, that's the reason people are interested in giving more anti-inflammatory drugs, so that inflammation is at the base, is hampered, is very less, and then that will be like the doubling phase of cancers will go down, and the malignancy rate will be decreased. And going to the next question, as you have rightly asked, that this kind of okay. First, I would like to tell you about boron treatment. Yes, boron nanoparticles, like iron oxide, they are also very, very effective in the treatment of cancer cells. One is like they're responsible for hypothermia. And this hypothermia is something which is very, very effective in the treatment of, I mean to say, when the boron capture happens, and there is a beautiful mechanism of boron capturing in cancer cells, for sure. But there is, what happens is there is not one single treatment strategy still for this kind of cancer treatment. Like we can go for boron treatment. We can, even I'm saying that I cannot say, okay, I'm dealing with iron oxide or cancer, or sorry, gold and iron oxide, or if I like, even I have been working with boron. But there is some problem which we could find was the targeted delivery, number one, effective killing of all the cancer cells. Mostly the problem is like we are trying to kill the cancer cells or cancerous tissue, tumor tissue, but 100% killing is something which we are still not like we cannot do. Why 100% killing is not happening? Because first, as I mentioned, the detection of like the cells is not happening. The detection of cancer cells is not happening at the very, very low numbers. If the numbers are like in 10s or 20s, the numbers are like, those numbers are very difficult to be detected. And that's the reason we do the killing, and then we feel that, oh, come on, the killing has happened, not a problem. But then in the next few years or 1 year, even within 1 year, like there are left out residual cancer cells which might have been there, or there are stem cancer cells which are also there, cancer stem cells basically, they're also there. So we have not killed them, and they will just do the differentiation and they will be doing the havoc. So this is something wherein everybody is working still. And I forgot the third question, doctor. I am sorry. Sir, I'm very sorry.
The next question was use of interferons.
Interferons, yes. Because interferon gamma, interferon beta, these, and especially interferon gamma is a treatment strategy. So nowadays people are, of course, not nowadays. It's like Baltimore. He got the Nobel Prize for Temin and Baltimore. They got the Nobel Prize for interferons. From that age, interferons have been used for, and even tumor necrosis factor alpha, they have been used. But there is a big but. The big but is that interferons are in, like, they were giving as in the form of a drug previously. Interferons were given in the form of a drug. But then very recently we could find that interferons, they induce inflammation, and they induce macrophages, and those, and especially negative macrophages, which can help the cancerous cells. So this kind of study has come up in the year 2002, 2001, 2002. People have started working. They have started imagining, oh, come on, till the age of 90, like when Baltimore was studying, on Temin was studying, they could find, they have done certain studies wherein they found that, come on, when we give interferon gamma, the treatment happens and the cancer cells get killed. But then in the year 2001, 2002, when there were other scientists working on interferon gamma especially, and they could find, come on, this induces inflammation. And then there is altogether a different line of like the cancer cells, they take a U-turn basically. Whenever interferon gamma is injected in the body, they completely take a U-turn. And there is an inflammation happening in the system. So this interferon gamma is like a double-edged sword, which has become it's a double-edged sword which is creating problems. But yes, interleukin 6, interleukin 4, interleukin 10, these are all anti-inflammatory drugs, anti-inflammatory cytokines, along with interferon gamma. But interferon gamma, tumor necrosis factor alpha, these are the ones which have to be used with a very, very with the utmost care. So it's not that effective treatment for cancer, for sure. Yeah. So that's, and even we are trying to do that, because in breast cancer on a chip, we're also using different kinds of cytokines, especially interferons and interleukins. But in my study, interferons are doing well. But I could find this kind of study in the human patients, and interferon gamma is not doing a great job. So that's a big problem nowadays. Yeah.
Thank you so much. Thank you so much.
Thank you. Thank you, sir. Thank you.
Okay, good morning, sir. Dr. Jagriti here from Chemistry Department.
Hello, ma'am. Hello. Nice to meet you.
Sir, first of all, congratulations for a wonderful talk. Sir, I have one question regarding the amount of folic acid used. Is there any restriction to use the amount of folic acid? Because if you are using it in excess, definitely it is going to have many side effects. So, what criteria we are choosing to decide the concentration of folic acid?
I could not understand the question because folic acid concentration, in the sense, I could not understand this point.
Sir, when we are using folic acid as a receptor, so how much folic acid we are going to use?
Okay. Madam, actually, we are not using folic acid as a receptor. Folic acid is a ligand. And folic acid receptors means the receptors, it's a protein, right? Basically, right? It's a receptor is a protein. So the folic acid is a ligand, and there is a receptor which will interact with each other. So and now when we are using folic acid onto the surface of nanoparticles, are you asking that question?
Yes, sir. Yes, sir. Yes, sir.
Thank you. Yeah. So folic acid, when we are using onto the surface of nanoparticles, they are used, first of all, the concentrations of nanoparticle itself is like 50 microgram per deciliter. That is a concentration. And when you talk about folic acid, yes, folic acid, the ratio is 1:10, like 10. Like the nanoparticles will be 1 and 10 will be folic acid. So, you can imagine like 50 microgram means 15 to 10, 500 micrograms, that much. Like that is the number of concentration. Sorry, that is the concentration approximately we are using onto the nanoparticles. So when we are talking about these folic acid, this folic acid which is interacting with the nanoparticles, which is already on the nanoparticles, this whole complex will motivate the nanoparticles towards the cancer cells. That's the only job. Rest, folic acid doesn't do anything else. They will just motivate the nanoparticles. They will so that there is a synaptic delivery or a targeted delivery of the cancer cells. I mean to say delivery of these nanoparticles into the cancer cells. Nothing else. So even if folic acid concentrations are higher, okay, so then also these folic acid will be, either it gets into other cells, which is very good, or they will, if at all they are getting into the cancer cells, yes, they cannot get into the, for sure, they're with my studies, I could find they're not getting into the normal cells, of course, because the normal cells have got almost 10 or 10 raised to 2 folic acid receptors. We could through flow cytometry, we could also measure the number of receptors. So we have actually measured what is the number of receptors in cancer cells and what is the number of folate receptors on normal cells. So we could do that kind of measurement, and finally we could find that, okay, even if we have 10 or 10 raised to 2, my nanoparticles are not entering inside the normal cells at all, and this can be seen in the cytotoxicity of the normal cells. The normal cells, hardly there is 1 or 2 cells getting killed, but in the case of cancer cells, like there are 10 raised to 5 cells or 10 raised to 6 cells getting killed using through the methodology of apoptosis. So that's something which is, so the number of folic acid is, even if it is higher, it's not a problem. They will be moving, the receptors are important for them, not the folic acid. So if the receptors are there, they will enter. The receptors are not there, they will not at all. They will never enter.
Yes, sir. Thank you very much, sir. Thank you.
You're welcome.
Excuse me, sir.
Yes.
Hello, sir.
Hello.
Yes, sir. I have a question. Is there any way that we can convert the non-malignant, that means the malignant cells that are the cancer-causing cells, into the non-malignant cancer-causing cells?
I'm sorry, if you can be a little louder.
Whoever it is, please introduce yourself and be a bit louder. Hello.
Hello.
Yeah. Pratibha.
Hello. Yes.
Yes, tell
Yes.
Pratibha.
So my question was, is there any way that we can convert the malignant cells into non-malignant cells?
Okay, her question is, sir,
Mhm.
non-malignant cells can be converted to malignant cells can be converted to non-malignant cells.
That's
Is
a
it
question.
possible? Yeah, is it possible?
Okay, yeah, that's something which is like basically there are certain plant extracts which have been studied, and those plant extracts are like Vinca, and there are certain other kinds of extracts which are partially converting a malignant to a non-malignant, but this rate is almost like 0.01 to 1, not 0.01 to 0.1. That is the rate. So there is like this conversion rate has not happened. Okay, why? What is the reason? First of all, the converting malignant to non-malignant is not important. What is important is we need to activate the immune system. The biggest problem in a cancerous patient is immune system doesn't work effectively.
Okay.
There is a patient, actually we have been like she's a model for us basically. So she has got breast cancer. Next, she has got liver cancer. Third, she has got stomach cancer.
Okay.
Generally, what happens is all these three different kinds of cancers will happen. Now, what you will think? You will think, okay, come on, it's like it's a primary tumor. First, she got breast cancer. Then the primary cells must have, like the primary tumor cells must have come out from there, and then they must have attacked the liver cells. Then there is like a secondary tumor generated over there. Then the cells must have come out and reached to stomach. No, this has not happened. What has happened instead is there is a primary tumor which is developed in breast. So she got breast cancer. There is a primary tumor developed in liver. She got separately liver cancer.
Okay.
And then she got a separate stomach cancer, all three different cancers. So she has become a model for these doctors, oncologists, basically. Because so then they started studying the genomics. So through the genomics part, they could understand that all the her like whole family suffered from cancer. First of all. Second point is immune system. There is a defect in the immune system, especially in the macrophages. And due to the defect in the macrophages, they were not easily killing. I mean to say, the killing of cancer cells, like the detection of the cancer cells and the killing of the cancer cells and the induction of apoptosis is not happening. So then immune cells are creating problems. If the immune system works well, then they can easily kill the cancer cells. All the malignant cells will be killed. Okay, because see, if anything, any part of the body is damaged, if the cells are damaged, so we need to remove them. That's the best option. We don't have to repair, like cells, like the body does the repair work for sure, but the best option is to kill them, to remove them from the body as early as possible. And body does that. Like normally our body does that. If there is a normal person, it happens. But in the case of a cancer patient, this doesn't happen so easily. So they are accumulating the cells, immune system doesn't work properly, effectively, and the killing doesn't, and the killing doesn't happen, and then finally the person dies because of cancer. So the most important aspect is not just conversion of malignant to non-malignant, but effectiveness of the immune system is very important.
Right, right. Yeah. So that was anyone else who would like to ask any queries?
Yes, ma'am. Can I ask?
Yeah, sure.
Please,
Go ahead.
please.
Please introduce your name and then give the question.
Sir, my name is Sagar Gupta from Zoology, TYBSc Zoology. Sir, I am asking that is there any genetics about cancer? Is there cancer have any genetics?
Yes, for sure. As I'm telling you, there are certain proto-oncogenes, and I think you must be aware of. There are certain proto-oncogenes and oncogenes, right? So these proto-oncogenes, we all have proto-oncogenes with us, but that conversion of the proto-oncogenes to oncogenes, that regulation, there is a regulation, and that regulation, if there is a mutation or if there is any genetic basis, like parents or the forefathers, anybody has got cancer, then the chances for the children to get cancer becomes like at least 20 to 30% higher. So the thing is, we should not be afraid of it. The most important aspect is the best option, and the most important aspect is that how we can. It's basically our day-to-day life, right? Nowadays, the life. I mean to say, the lifestyle has become very bad in all senses. So if that 20 to 30% can be enhanced to 40 to 45% due to the bad lifestyle, and if there is a good lifestyle, that 20 to 30% can go down to 5 to 10% also. So this kind of thing happens only due to a very, like, the kind of lifestyle you live, the stress-free, then the body and the sleeping orderliness. So all these aspects are very important for cancer, for sure. And the kind of food which you're eating, that's very important also. So yes, it is genetically, it's very important. And the most important is aging. So as aging happens, like 60 years, 70 years, you will find that mostly like patients are above 60 to 70 years old, at least 60 years old, mostly. So due to the aging, there are like telomeres, and they are playing an important role, and that's the reason this telomerase inhibitors are available in the market. So these are very important aspect for sure. Genomics is surely important. Yeah.
Sir, can I ask one question on?
Please.
Sir, if a pure person if he is done his health checkup a day before, but he sees cancer symptoms on next day itself, is it possible?
I'm sorry, I could not understand. Tell me again.
Sir, if a pure person, as if he has done his health checkup a day before, but he sees cancer symptoms on the next day itself, is it possible?
What kind of health checkup he has done? Okay, I will tell you. Don't worry. Yes, I understood that point. That's a very common thing nowadays. All the, like, if you take the reports of cancer patient, everything will be normal. Everything will be normal. Her like CRP levels are normal. CRP, which is considered to be inflammation marker, that will be normal. C-reactive proteins. Hemoglobin will be normal. All erythrocytes number, everything will be normal. All right, but then also the patient is suffering from cancer. So the blood report is not, there is no relationship between a blood report and the report, and the cancer patient. Because I have so many reports, like whenever we are working on breast cancer, we need to get the complete report analysis. So and I'm totally amazed by that. Okay, which particular marker helps me to identify that the patient is suffering from, that the person is suffering from cancer? Which particular marker? So we are trying to do that. So we did this machine learning strategies. So through the machine learning strategies, we could find there are 6 to 7 markers, which we are not even testing nowadays. We are not even testing them in the normal blood reports, not at all. So now in future, those 6 to 7 markers might also be introduced. Like, for example, if I want to confirm that there is a patient who is triple negative breast cancer. So for that triple negative breast cancer patient, these are the 6 markers we need to do. If these 6 markers are not done, whatever blood checkups you're doing, it's a waste basically. So this is something which is very critical. So just don't believe on the normal blood reports. So whatever blood reports you're doing, it's useless. You cannot confirm that, okay, this patient is normal or abnormal or having cancer using this blood report.
Okay, thank you very much, sir. Saga, that was good questions. I think we are running out on time. Yeah, anyone else?
Hello, sir.
Hello,
Yeah.
Utkarsha.
Myself,
Hello.
Assistant Professor Utkarsha Dhuri from Zoology Department.
Hello.
Actually, I want to ask about the role of telomerase enzymes in cancer treatment, because as we know that there is a theory of telomerase shortening theory in aging and cirrhosis. The stability of the genetic material gets lessened by the age because of less amount of telomerase, So what exactly we are doing, or whether we are targeting the telomerase in the cancer treatment, and if it is so, then what kind of agents or chemicals we are using for targeting that enzyme? That I want to ask about.
Actually, there are certain drugs like Vivacisumab, and there are certain drugs which will be acting against the telomerases. And then if the telomerases are inhibited, then the aging factors, like the aging process, I mean to say, this aging process basically is hampered. Not the aging process. Basically, these cancer cells, if the telomerase is inhibited, then there is induction of apoptosis in the cancer cells. And if there is induction in the apoptosis of the cancer cells, then they are this is the mechanism. There is an indirect mechanism of killing cancer cells. So that's the reason they are doing inhibition of telomerases. So this is the way they are the patients are being treated.
Thank you so much, sir.
Thank you.
Yeah, thank you, sir. Thank you, ma'am. Anyone else to ask for questions? Okay. So, sir, we are almost towards the end. It was a brilliant session. Now we'll go ahead and we are about to get to the conclusion. So this brings us to the end of the session. So may I call upon my colleague, Ms. Utkarsha Dhuri, to give a vote of thanks and end the session. Utkarsha, ma'am.
Am I audible to everyone?
Yes. Yes.
Thank you so much, Nisha madam. Hello everyone. As all good things come to an end, so is the webinar. On behalf of Zoology Department of KV Pendarkar College, Autonomous Dombivli, I, Utkarsha Dhuri, take this opportunity to thank to those who have directly and indirectly supported and contributed to this international webinar. I would like to express my sincere gratitude to our Honorable Chairman of Dombivli Shikshan Prasarak Mandal, Shri Prabhakar Desai sir, for giving us the opportunity to organize this webinar. We are thankful to our guiding force and mentor behind this event, Shri Vinayak Dalvi sir, for his constant support. I would like to express my gratitude to our esteemed chief guest, Professor Goldie Oza, to take out time for his
busy schedule to enlighten us with a valuable talk on breast cancer on a chip, a paradigm for studying breast cancer physiology for therapeutics. Thank you so much, sir, for your brilliant talk. Definitely, sir, you have explained the use of nanotechnology for the drug delivery in the cancer treatment. So, in very easiest way, I'm sure that everyone present here could understand the mechanism of the drug delivery in the cancer treatment, right from the formation of that nanoparticle, core cell nanoparticle, then the delivery of those particles towards the target site through various methods like macrophages. Then there is a generation of the microchip chips for the experimentation to study the drug delivery or the drug testings on the cancer treatment. So it was very great talk, and so thank you so much, sir. We are really enlightened with your knowledge and presence here. It is also our great pride to be associated with the Goldie sir, because we have several alumni students who have registered under his guidance for the PhD as well. So thank you so much, sir. Now, I would like to thank our respected principal, Dr. Suryakant Lasoni sir, for his enthusiastic support for organizing this webinar. I also extend my gratitude to our senior vice principal, Mr. Falak sir, vice principal BT Shirsat sir, Mrs. Ashwini Bakar Madam, for their constant support and guidance. Special thanks are also due to involvement and contribution of our senior colleagues, Dr. Bholanath Mukherjee sir from Nanotechnology Department. A special thanks to organizing team, teaching and non-teaching staff for unflinching support and coordination for this webinar. Our heartfelt thanks to our students for their active participation, patient listening for this event. I would also express my special thanks to our technicians, Mr. Tahir Patel, for his technical support. So, with these warm words, we move to the end of this today's webinar. Thank you so much, everyone, for making this webinar grand success. So with this, I conclude that today's session is over now. You may leave the meeting at your own means. Thank you so much.
Thank you. Thank you.
Thank you, sir.
Thank you very much, sir. That was indeed
Thank
a
you very
very
much.
enlightening.
It must be midnight in the Mexico now, right?
Yeah, it's 12:30. Yeah. No,
Yeah.
not a problem.
A lot of pains
must
you
be
have
really
taken
tired.
for all of us.
Yeah.
It's my
Take
Yeah,
care,
thank
sir.
you.
and
Thank you very
I
much.
would like to I will be very much happy in the near future also to be a part of this kind of sessions, and in the very near future as well. And if there is anything which I can help, I would be more than happy to guide the students as well as guide professors. Thank you. Thanks.
Thank you
Thank
so
you very
much.
much.
Grateful
Thank
to
you.
you, sir.
Thank you, sir.